Welcome back to my YouTube channel. For those who are new here, karibu sana. I'm so glad to have you. Welcome on board. So today we're going to do a more of an educative video, as this channel is more of fun, entertainment, and educative videos. So today we have one of those educative videos. So karibu sana. My name is Rehema Liai. So our topic today is testing of COVID-19, and I'll just want to go through a few things about how we test for COVID-19 in the laboratory. And first of all, we just need to understand what is COVID-19. So COVID-19 is just basically CO stands for corona, VI stands for virus, and D stands for disease. 19 is the year that the disease was discovered. So COVID-19 is a viral disease that was discovered in Asia, and we call it a zoonotic disease because it's basically transmitted from animals to humans, and once it gets to humans, it can also transmit among humans. So COVID-19 is very closely related to MERS and SARS. These are other viral infections that have been there before, and the normal incubation period is about 2 to 12 days. That is like average days for the incubation of the COVID virus in the human body. And this is the reason as to why quarantine is for 14 days, because within that period, it'll be able to replicate in a human body, and it can also be tested. So what are the symptoms for COVID-19? As we all know, or as you've always been seeing in all this TV adverts, all the posters wherever we go, the main symptoms are fever, cough, nausea. You can get some congestion, you can get some diarrhea, depending on how severe it is. So there are a couple of tests that are done to test for COVID. And the main aim of this video, actually, I would just like to explain in the most, I'm trying to look for a word, in the most simplest way possible, without using a lot of big scientific words, on how we basically test for this virus. Yeah. So snapshot: COVID, coronavirus disease, 2019. One thing that is unique for COVID, it causes both upper respiratory tract infection and the lower respiratory tract infection. Okay, so the first thing they have to do is a sample collection. But before you collect the sample, since we know that this virus is highly infectious, the personnel or the health worker who collects this sample definitely has to protect himself or herself. So basically, we put on the Tyrovac suit. You have to wear the whole thing, and then you have to put on your glasses, and then you have to put an N95 mask, and then put on the whole shield, because you don't want any part of your body exposed. And this is the swab. It's that long. Of course, it is sealed. It's not contaminated or anything. So it has instructions on how you open it. I'm just going to open this. This is how the swab looks like. This part is more, it's just soft, like, you know, little tiny sponge, and it has like demarcations. Yeah. So let's see how long this thing is if I measure it on my arm. So yeah, that is about how long it is. Yeah. So basically what is done, you have to be at some sort of an angle, and this will be pushed like like deep inside your nose. So I will do the very first step of taking the nasal swab, and then I'll explain everything after that. Testing for COVID, how does it start? So first of all, a lab technician has to collect sample from the person who has exhibited these symptoms, or if you just want to know your status.
Swap. Do you have any problem with allergy like chronic rhinitis, chronic nasal blockage?
No, just a normal pollen allergy.
Pollen allergy. And a history of hospital?
Yes.
One of the last hospital?
Like a year.
A year.
Yeah.
Was it treated?
Well, I think I just said this because I was still growing up. Like this used to start on its own even at night.
So I want to take a nasal swab. You want me to do it on the left or right? So can you slide the tip your hand backwards? Don't pull my hand, please. Yes. Careful. Just sit down there. Is there any pain? 1, 2, 3, 4, 5. Now take it down like so. And I'm through. Yeah.
Oh. Guys, it's done.
What next?
Okay.
How was it?
It's not as bad.
Yeah.
It's just quite uncomfortable, but yeah.
Yeah, no pain. No?
Yeah. That's it, guys. So I'll explain the next procedure after.
Thank you.
Okay, thank you.
See you.
After that is collected, usually they will cut. They will have to like break off some part. Yeah. And whatever remains, they have this small part that they will put inside a small tube. Usually in this tube, it contains something called the VTM, that is a viral transport media. Once you cut off this swab and you put it inside and just like close it in properly. Usually when working with science, you always open the tubes with your left hand because this hand is holding your pipette, right? So you open the tube, yeah. Once you open it, you're going to use your pipette to suction the fluid from inside to a certain volume depending on the protocol that we're using. So you get the volume and then you don't want this to spill on you, so you seal it. Seal it very well and put it aside. And then this, we're now going to transfer it into this. This is called an Eppendorf tube. And that is where the downstream analysis begins. So all we have to do is to extract RNA from that sample that we got, because when we extract the RNA, that is only when we can tell whether this person is positive for COVID or not. So usually we use a couple of reagents. One of them is called the proteinase K. This is the proteinase K. So what the proteinase K does is that it's going to break down the proteins inside the sample that we have. And then next, we're going to add the lysis buffer. So the lysis buffer is basically going to break the cell. So you know, like the human body, we have this. This is the cell. So say this is the, the virus cell. So inside we have the RNA. So for us to get into the RNA, we need to break this cell, like so that the RNA will be exposed. So that's why we use the proteinase K and the lysis buffer, so that once this RNA is exposed, still in the fluid, this all happens, Once it is exposed, we're now going to move that into another column that is called the spin column. I'll just show you how the spin column look like. So this is the spin column. It's detachable. So again, with your left hand, open it, move the fluid from this tube where you added your protein SK and your lysis buffer, and then put it in here and then close it. So a little science of what happens here. This tip has more like it has some sort of a filter. I'm not sure whether you can be able to see it, but it has some sort of a filter in there. So when you put it like this, we're going to centrifugate the fluid that was in that Eppendorf tube, or in this small tiny tube that you put in here. That fluid is going to pass down and go in here. But whatever you remain with will be your RNA that will be captured in here. It will not sieve through, it will not pass through. So with that, we then now go ahead and do what we call washes. So washes is just using isopropanol or ethanol, depending with what protocol we are using to do the wash until we get our RNA. So now we have our RNA. Still, it's just a very small, tiny, colorless fluid that it's not like you're going to look at it like this and see RNA. So the next step that we do from there, we're going to do something that is called PCR. PCR is a short form of polymerase chain reaction, and in simple terms, this is photocopying. We have a little amount of viral RNA and we want to make more copies. So the way you can go to a cyber cafe and photocopy your documents and have like hundreds and hundreds of them is the same thing that we do for this sample. So we put it into the PCR machine and it's going to make more copies of this RNA. And while doing this PCR, we also have to add some other reagents that are going to amplify, looking for very easy words, that is going to identify this RNA, like e-viral RNA in that sample. Once a sample is positive, if a sample is positive, the PCR machine is going to pick out an RNA for this virus, and then it will show you, you'll have like curves. You'll see like a blue, green, white, depending on what color you set, but you will see like a green or 3 curves that will show you that this sample is positive for corona. And if you don't find that, you'll see that this sample is not positive for corona. And yeah, so basically that is what we do. Oh, and the other thing I forgot, when we're also doing this PCR, if you want to know whether it's a human sample, there's also something else that we target. There's a human gene that we target, and then we have like 2 other genes that we target from the viral RNA. So in total, there are 3 things that you target. So all these 3 things, they will show you different 3 curves on the machine, the PCR computer machine. So when you see all these 3 curves, so that means the human gene has been amplified, meaning the human gene was present So it has been photocopied. And then one gene from the virus has also been amplified, and then another gene from the virus has also been amplified. So when you see a sample with 1, 2, 3, the 2 genes from the virus and 1 gene from the human that have been amplified, then that is a positive sample. Then if we get only one gene that has been amplified, like you just get 1 curve from 1 sample, or if only the human gene has been amplified, then this is a negative sample, because the 2 genes that were not amplified or they haven't been detected in the sample means it wasn't present. So corona was not present in the sample. That is a negative sample. So I don't know whether this was clear, but yeah. So anyways, 2 things why this takes quite a lot of time. Extracting the viral RNA takes quite some time. It can take a whole day depending on how many samples you're handling. And once that is done, we again now go to the PCR. The PCR is not just like a 5-minute thing. On an average, it takes about 2 hours for you to get the results from the PCR machine. So DNA or RNA extraction, and then PCR, and then you have to compile the results, and that is why you get your results after about 24 hours, because that's the process in the laboratory is quite a task. Yeah, that's it for today, guys. I hope you liked and enjoyed this video, and yeah, kindly remember to always sanitize. Remember to sanitize, guys. Sanitize always. If you can't sanitize, just wash your hands with soap and water. Always use your mask every time you're out, and keep the social distance, and stay safe. And until next time, peace out, guys.